Hey everyone, welcome to Chapter 2, Part 10, our final piece of the neuro unit. This time we're talking about cerebral cortex. So your cerebrum is the two hemispheres containing cerebral cortex and underlying subcortical structures. Subcortical structures just being those structures that are located below the level of the cortex. This can be divided into four distinct lobes, which we're going to talk about in more detail coming up. Cerebral cortex is the thin layer of interconnected neural cells covering the cerebral hemispheres. Cortex means rind or bark, so it's sort of the outer covering of our brain. Larger cortex is related to increased learning, thinking, and adaptation capacity. So generally speaking, the more sort of sophisticated an animal is, the more sort of large and convoluted the cortex will be. So cerebral cortex, this is the outermost layer of gray matter of the cerebral hemispheres. Remember, gray matter is cell bodies, and white matter is the axons. So this is the layer of cell bodies that is the outermost layer of our cerebral hemisphere. A sulcus is the groove in the surface of the cortex. So little indents right here, the sort of valleys between the peaks, which we call gyrus. Gyri are convolutions of the cortex. So the sort of mountains, the sort of protrusions are gyri, and the indentations, the valleys, are sulci. So the frontal lobe, pictured here in red, is most important for speaking, muscle movements, planning, judgment, and decision making. This contains the motor cortex right here, which is something else that we'll talk about in more detail coming up. So Phineas Gage is a very interesting case study. He was a railroad worker in the 1800s who suffered a horrible brain injury when he inadvertently blasted a steel rod through his left frontal lobe, destroying a significant portion of this part of his brain. Miraculously, he survived the incident, but he was changed. He survived, but was no longer Gage. Before the accident, was known as kind and soft-spoken. He became profane and dishonest. Frontal lobe is really, really important for sort of top-down control, planning, emotion regulation, all these sorts of things. So despite surviving the accident, here in yellow is important for integrating sensory information, knowledge of numbers and their relations, manipulation of objects, and spatial vision. This also contains the somatosensory cortex, which is located right here. Okay, so I want to talk a little bit about the interesting and unique organization of the motor and somatosensory cortices. So pictured here in purple is our motor cortex. This is the part of our brain that's really important for initiating and guiding voluntary movement. You can see here that there's been sort of a cartoon person mapped onto the strip of cortex. That's because it has what we call topographical organization. It's very reliably organized in a fashion which a specific portion of the cortex controls a specific portion of your body. And the amount of real estate that body part occupies on the cortex is a function of how much fine control we have over it. So something like our face or our hands takes up a lot of space, despite being relatively small compared to something like our trunk or our hip. We just have more fine-grained control of those parts of our body. So more brain space devoted to moving it. Likewise, the somatosensory cortex is responsible for the processing of sensory information. So things like touch and temperature, etc., from our periphery. You can see here there's a very similar organization where things like our face and our hands, which have are much more sensitive than other parts, have a lot more processing power devoted to them. Things like our neck and our trunk, which are not very sensitive, take up less space despite being comparatively larger. We just have a more dense array of receptors for things like our face and our hands than we do for like the back of our knee, for instance. The occipital lobe, pictured here in purple, is our visual processing center and is our smallest lobe. And finally, our temporal lobe, pictured here in teal, is important for auditory processing, memory, emotion, and language comprehension. So we have a large amount of our cortex devoted to association areas. These are found in all four lobes, and they're important for interpreting, integrating, and acting on sensory information and allowing it to be linked with stored memories. This is basically any portion of our cortex that's not concerned with the primary motor or sensory functions. So we have parts of our brain that we consider primary cortices that are doing the initial processing of these things, like motor cortex initiating our voluntary movement, or our primary visual cortex and our occipital lobe doing the initial processing of visual information as it's coming in. Anything that's not a primary cortex is an association cortex. So for example, our frontal lobe association cortices allow us to engage in high-level stuff like judgment, planning, and processing of new memories. All right, let's talk very briefly about some imaging methods that are used in humans. First up is EEG, or electroencephalogram. This gives us a readout of the brain's electrical activity and gives us information about function of what's happening. So EEG allows us to basically take advantage of some of the properties that we've talked about. Remember, initially we talked about how when neurons talk to one another, they undergo these changes in electrical activity where ions are moving in and out of the neuron in a predictable way. You can imagine that that's going to cause large changes in electrical activity that can be read along the cortex. So what EEG does is places sensitive electrodes on the scalp surface, which are sensitive enough to pick up those sweeping changes across populations of neurons. So this gives us a readout of real-time sort of what brain regions are active and in what way. The really interesting thing about EEG is it's extremely temporally specific. So it gives you a lot of information about when things are firing on like a millisecond level of resolution. PET, or PET scans, positron emission tomography, these measure basically brain metabolism, right? So you would administer some radioactive glucose and you would watch where the signal of that radioactive glucose decaying is coming from. Now, that sounds dangerous, but it's harmless radiation. So basically, with a PET scan, you can tell what parts of the brain are taking up this radioactive glucose and using it. That's sort of an indirect index of how hard a brain region is working. The more activity that's taking place in a particular brain region, the more glucose it's going to be using. So it's an index, basically an indirect, sort of slow index of what part of the brain is most active. This gives us some information about structure and function, so you can see where neurons are and where that activity is taking place, so how much activity is taking place in a specific structural location. So fMRI, or a functional magnetic resonance imaging, takes advantage of some of the properties of basically blood flow. It can use the magnet to visualize the location of oxygenated. So fMRI, or functional magnetic resonance imaging, takes advantage of some of the magnetic properties of oxygenated blood and can visualize, using a very powerful magnet, where blood flow is going. With that, we can get sort of an index in sort of a similar logical vein to PET scans with fMRI. We can learn what brain regions are using more oxygen than others, right? An active brain region is going to be using more oxygenated blood. So by using those the magnetic properties of oxygen, we can visualize what areas of the brain are more active. So we get information about structure, the same we would with MRI, and function. We get to see what brain regions are most active. An additional technique is called diffusion tensor imaging. This visualizes the movement of water along axons and can provide us information about basically projection pathways, where a population of neurons is sending its axons by visualizing the direction that water is moving along those axons. Okay, just to summarize again very briefly, EEG provides really great information about functionally what's happening in terms of neural activity, less detailed information on structure. You can't localize the signal with the EEG quite as precisely as with some other methods, but with dense arrays, it's better. So we can get varied amounts of resolution based on how fancy the setup is. PET scans provide sort of crude metabolic information. You can tell about where the signal is coming from. It's of course delayed because we're looking at glucose metabolism. MRI shows us structural information about the brain, and fMRI shows us that same structural information, but also metabolic information about how much oxygen a particular brain region is. So speaking of axons communicating information, I want to mention the corpus callosum, which is a large band of axons that connects the corresponding parts of the association cortex on the left and right hemispheres. So it's this thick bit of white matter right here that serves as sort of a bridge connecting the two hemispheres. This allows our two hemispheres, which have some degree of specialization, to communicate easily with one another. So, given how important the corpus callosum is in allowing our hemispheres to communicate, you might wonder what would happen if our brains could not communicate so easily hemisphere to hemisphere. Well, there is a small population of patients that suffer from severe epileptic seizures, and in order to treat these seizures, the corpus callosums have been severed. This did, in fact, alleviate the symptoms of seizures, but there were some large consequences. So I posted a video of a case study on somebody who's had this procedure. You can find that on perusal. So make sure to watch that for a sort of a deeper dive into exactly what happens with a split brain patient. Okay, that wraps up our neuro unit, and I'll see you next time.